Welcome to everyone joining us, and I'd like to introduce Dr. Nayana Lahiri. And Nayana is a consultant clinical geneticist working at St George's in London. And Nayana is going to talk today about what you can see on the screen there, everything you need to know about the genetics of Huntington's disease.
As John said, I'm a consultant in clinical genetics at St George's Hospital in London. And I've been working with HD families for about 15 years now, both in research and also in the clinic. And so what I'm going to try and do is to give you a little bit of a reminder about what a gene is, how it makes proteins, and its sort of inheritance of Huntington's disease. To try and explain what the CAG triplet repeat means and what it stands for, to talk about how genetics can help to explain why Huntington's disease can be quite so variable, to think about the difference between a diagnostic and a predictive test, to explain some of the options for family planning that involve genetics, and also thinking about how our current knowledge of the genetics of Huntington's disease is leading to development of potential therapies for the future, and then hopefully to point you in the direction of where you might be able to find out more, because that's quite a lot to cover in 45 minutes to an hour. And so there's always options for further reading if you are interested. So really, Huntington's disease is a family condition, and you all may have seen families who have been affected with Huntington's disease. That is why you are probably here. But it is really important for us as clinicians when we're seeing families to sort of understand how the Huntington's disease affects not just one person often, but many people within a family, not only those who are affected, but also those who are caring for people who are affected. And sometimes it is also really important to remember how variable the condition can be, and the impact of making a diagnosis on one person, what that has on the rest of the family. This is Charlotte. She is a young lady who has sort of shared her story through the BBC Lifeline appeal with Huntington's disease this year, and I'm going to sort of use her as an example of how HD can affect families. And if you haven't seen her video and her story, then I would highly encourage you to go and have a look at the website and watch her videos. Really, so just to start off with some very basic biology, taking you back to your GCSE days, all of our bodies are made up of thousands and billions of cells, and the fundamental, they're the fundamental building blocks of how our bodies work and how our bodies function, and the cells are important for making sure that different parts of the body do different things. And in the middle of each of our cells, mostly, is a nucleus, and within the middle of that nucleus is where all of our genetic material is stored. And so largely in all of our cells, we have a full, complete amount of our all of our genetic material that are stored in structures called chromosomes. And but the lengths of DNA are made up of genes, and each single gene will be transcribed to make RNA, and then the RNA will be translated to make amino acid chains, and those amino acid chains then form structures called proteins, and the proteins are like perform all of the functions in the body. And so the protein that we're talking about that is affected in Huntington's disease is a protein called Huntingtin. So what does Huntingtin do? Well, you might read in some textbooks and people might tell you that we don't know what it does, but actually that's because it is a complicated protein and it does lots of different things. So Huntingtin is a protein that is expressed almost in every cell of the body, and it's really important for helping transport other proteins around. It's important for controlling how cells signal other cells to do other things, and it's really also important in controlling the regulation of other genes. And so there are lots and lots of different functions in cells that it is important for. It is particularly important for a healthy brain for cell function, and it is particularly expressed in certain regions of the gene, which are called certain regions of the brain, which are called the basal ganglia. But it is there and present and being made in lots of different cells doing lots of different things. And so what causes Huntington's disease? Well, it's when something happens to the genetic material that then causes the protein not to be made completely normally. And the reason that so Huntington's disease was described in the 1800s, and it was understood to be a family condition at that stage, but it's really only sort of through the work of a number of different scientists and an international collaborative effort in the 80s and the 90s that the gene for Huntington's disease was discovered. And largely that is due to sort of looking in a population in Venezuela where there were lots and lots of families who were affected, and they were hugely important in trying to identify the gene. And what we've always known about from looking at families is that Huntington's disease, even before we knew what the gene was and what it did, was that it was inherited in what we call an autosomal dominant fashion, which means that if a parent was affected with Huntington's disease, so they had then they would be a 50-50 chance for any children of also being at risk of becoming affected with Huntington's disease. And so these are the cells of the parent. You can see that the red copy is the changed copy of the gene, and the white copy is a normal copy of the gene. And so when that parent comes on to make their eggs or their sperm, half of the eggs and half of the sperm would carry the changed copy of the gene, and therefore there'd be a 50-50 chance for any children of inheriting Huntington's disease from them. So through gene discovery and through investigating families, so this is what all of your genetic material looks like under a microscope. So these are the chromosomes. They are labeled in size order from the biggest ones, which are chromosome pair number 1, to the sex chromosomes, which are the X and the Y chromosome. And you can see that our chromosomes come in pairs, so we've got 2 copies of every chromosome, and it's through that sort of gene discovery work in the 1980s that we identified that the HD carrying chromosome was chromosome 4. And you can see that everyone has 2 copies of chromosome 4. I like to sort of think about all of your chromosomes being all of your genetic material being like the library, each of the chromosomes being like a bookshelf in that library, and each of the genes being an instruction manual for a protein that sits on one of those bookshelves. And this is what the HD gene looks like when you look at that instruction manual. So you can see that all of our genes are made up of 4 different letters, A, G, T, and C, and they are the building blocks for proteins. And the way that they do that is just from that 4 combination of genes, you can have lots and lots of different combinations of letters. They are read in 3s, and each collection of 3 letters codes for an amino acid, and that is the smallest building block for a gene. So you can see here at the very beginning of the HD gene, there is an A, C, A, and that encodes for an amino acid threonine. And then here a bit further on, there's a G, A, and that encodes for an amino acid called glutamic acid. And so it's this bit of the gene, quite near to the beginning of the gene, that is the bit that we are interested in Huntington's disease. as you can see here, there is a CAG, CAG, CAG repeated over and over again several times. And so CAG codes for this amino acid called glutamine, which is sometimes abbreviated to be a Q. I don't know why, but that's just what it is. It's only when, so everybody has a CAG repeat within that first coding region of the gene. So everybody has a repeat. It's only when that repeat becomes bigger than expected that would cause problems with this amino acid chain that then forms the Huntington protein. So everyone has two numbers in their genes, one on each copy of chromosome 4 for the length of their CAG repeat. It's only when that repeat is expanded above the normal level that you will be at risk of developing Huntington's disease. And sometimes, because there are other conditions that also have this repeat structure in it, they are collectively called polyglutamine diseases. So what does having an expansion do? Well, we know that having an expansion in the gene causes a change to the structure of the HD protein, and it can cause a number of different issues within the nerve cells and other cells in the body. This is MHTT is mutant Huntington, and we know that fragments of the Huntington can build up in the cells. They can cause toxicity to nerve cells, and that buildup of the faulty copy of the protein can then lead on to sort of neuronal degeneration and nerves not being able to repair themselves. And that happens because they clump up together in aggregates. And that's because when the protein is made, it doesn't fold quite as it should do to make the 3D structure of the protein, and then that in turn leads to alteration of the function of the protein, so it can't then do its normal functions, which is to sort of make sure that other genes are being transcribed properly. It also causes problems with the cell's machinery for getting rid of waste products, because it can sort of stick to structures in the cell that are supposed to be getting rid of other waste products, and it can also lead to changes in the way that energy is produced in the cells. So how do we do a genetic test for Huntington's disease? So this is Charlotte here with her grandmother and her mother, and her grandmother had symptoms of Huntington's disease and will have had testing for Huntington's disease. So a genetic test is just a blood test, and in Huntington's disease, it counts the number of CAG repeats there are on each copy of the HTT gene. So the result shows two numbers, and that tells us what the number of CAG repeats is on each copy of the chromosome. It doesn't tell you when you get that result when you might start to develop symptoms or what those symptoms might be, because everybody has their number of CAG repeats from the time that they are embryos, from the time that they're developing as babies. In the UK, diagnostic testing of Huntington's disease is carried out in the genomic laboratory hubs. So there are several across the country, and your blood sample will go off there, and it will be tested. And the testing differs slightly depending on whether you have a family history of Huntington's disease, or if you present to a neurologist with symptoms that could be in keeping with Huntington's disease. There are two different types of tests that we might do now. Sometimes people who don't have a family history of Huntington's disease might have something called whole genome sequencing, rather than just having a single gene test for the Huntington's disease gene. So it is important to think of a few things when having a diagnostic test for Huntington's disease. So usually the diagnosis of HD is made clinically. So we usually know that somebody has HD by examining them and by listening to the sort of their story of the symptoms that they've been experiencing, and also thinking about family history. But sometimes that is, and so sometimes it's a very straightforward diagnosis to make if there is a family history of Huntington's disease, but sometimes it's more complicated than that. It's important to think about what the test is and how long the results will take. So are we doing a single gene test for the HD gene, which usually takes about 6 weeks to 2 months to come back? Or is it a test that is much more broader than that through whole genome sequencing, which at the moment can take up to a year in some places to return? It's important to think about the person's knowledge of and experience of HD. So that's not only the clinician who is seeing somebody in clinic, but also the person who is having the test, because different people will have different perspectives on Huntington's disease and different amounts of knowledge about Huntington's disease. Being able to give people information in the way that they need it and understand it is really important. And also for the doctors, it's really important to understand the result that you are giving to somebody. It's important to think about sources of support. So not only when a diagnosis is made, but even while somebody is waiting for the results of genetic tests to come back. And it's obviously really important to think about the impact that a result might have on other family members, and try and help to prepare other family members and to prepare people for what that result might show. And so what is the result and what does it mean? So when we do a diagnostic test for Huntington's disease, or any test for Huntington's disease, in fact, the number of CAG repeats tells us something about the age of onset of the condition. So having up to 26 repeats in the gene is normal. And as I said, everyone will have 2 numbers for their gene. And so somebody might have 17 and 15, or somebody might have 17 and 20. So that would be a normal result for Huntington's disease genetic testing. It is only when one of those numbers is over 40 that you will be at risk of developing, will be at 100% risk of developing Huntington's disease throughout your lifetime. So more than 40 or more repeats is an HD causing gene change. And they're sometimes called full penetrance alleles. The higher that number is, in general, the earlier the age of onset tends to be. But it's a really variable thing. So if I saw somebody in clinic with 41 repeats, I couldn't tell them, well, you're going to develop symptoms at the age of 55. That's just not how it works. There's a huge range, and I can tell you a little bit more about that later. But I would be able to tell them, if you live long enough, you will develop symptoms of Huntington's disease. But the higher the number is, the earlier the age of onset tends to be. So over 60 repeats, we would usually expect somebody to develop symptoms in their teenage years, or sometimes even earlier. There are repeat sizes that go up to more than 100, and so juvenile onset Huntington's disease would be very likely in those people. But so you can see that between 26 and 40, there is a range that is not accounted for yet. So between 36 and 39 repeats, these are called reduced penetrance alleles. Somebody may or may not develop symptoms during their lifetime. The lifetime risk of Huntington's disease with 36 repeats is only about 1 in 4 people, or 25%. The lifetime risk of somebody with 39 repeats going on to develop Huntington's disease is much higher than that, so it's about 90%, but tends to come with an older age of onset, and that tends to also come with being a more slowly progressive condition. And so then between 27 and 35 repeats, these are not HD causing, so they do not cause symptoms of Huntington's disease. People with 27 to 35 repeats would be asymptomatic, but these are called intermediate alleles because there is a theoretical risk of passing an HD causing gene expansion to offspring. But they are present in quite a number of the general population. So around 6 to 8% of the general population carries an intermediate allele, and they are definitely not all expanding to cause HD in future generations. So there must be something particular about some of these intermediate alleles that can expand into future generations, and we're still working on what those things might be. So there must be other genetic and environmental modifiers for why that might happen in some families. So what we tend to see in generations of people who've been affected with Huntington's disease is that the age of diagnosis of HD tends to decrease through the generations. And there are a number of different reasons why that might be. It's called genetic anticipation. And it might be because if you know that you have a family history of a condition like Huntington's disease, then it's more likely that you would be looking out for symptoms in other family members, and it is more likely that you would be diagnosed at an earlier age because you were diagnosed at a time when symptoms might be sort of much milder and just starting to emerge because you're looking out for them. But it also can be that that CAG repeat size can change between generations. It could be that somebody in a future generation would have a higher CAG repeat than their parent, and that could also explain why the age of onset might be earlier. So these CAG repeats can be unstable, and that's because when you are making eggs and sperm, which are the the mechanism of making babies, you have to copy your genetic material over and over and over again to package into the eggs and the sperm for the next generation. And sometimes when you are doing that, the DNA replication apparatus can sort of add in a few or even remove a few of those CAG repeats, so it's not always perfect. And we don't understand why that is. And it is usually stable between generations, but there are some families in which it is unstable. And it might be something to do with genetic background, and I'll come on to that a little bit later. And it also can depend on the sex of the parent. So we tend to see that large expansions happen from a father to their children rather than from a mother, but it can happen in both. And I've seen families where there've been really large expansions from a mother to their children, but it tends to happen more in men. So sometimes people might ask, oh, well, I've heard in my family, I've heard that, girls can't develop Huntington's disease. That is not right. So whether you are a female or a male, there is a 50-50 chance of having inherited the HD gene from a parent. But where this male-female thing comes in is because it is just slightly more likely that there will be an increased expansion if you are a father and you are passing that on to your children. And what about when there is no family history? So we know, again, about 8% of people who are diagnosed with Huntington's disease don't have a family history. And that can be for a number of different reasons. It might be because somebody has been adopted, or somebody has, sort of been estranged from their family, or it might be because a parent has died very young, and so there wouldn't be, the family history wouldn't be unknown. It could be because a parent had died before they became symptomatic. It could be because there's a misdiagnosis in a previous generation. So sometimes people with Huntington's disease might have been mislabeled as having schizophrenia or mental health difficulties. It could be that they were labeled with having Parkinson's or Alzheimer's or another type of dementia. And so sometimes there might have been a misdiagnosis and no genetic testing had been performed in somebody who was affected. But it also could result as an expansion from a reduced penetrance or an intermediate allele, which means that somebody with Huntington's disease might have had a parent who carried one of those smaller CAG repeat expansions and hadn't necessarily become symptomatic during their lifetime. So what is predictive testing? Well, Charlotte's grandmother, she was diagnosed with Huntington's disease, and as was her mother. And then Charlotte has decided as a young person to think about having a test for herself to tell whether or not she's going to develop symptoms of Huntington's in the future. And so that is called predictive testing, where somebody who doesn't have any symptoms at all wants to think about having testing so that they can make plans for the future. And we have been able to offer predictive testing in the UK since 1993. And before testing was available, families with Huntington's disease in their family were asked whether or not they would have a predictive test if it were possible to do that. And overwhelmingly, people said, yes, we would like to have the option of having a genetic test. But actually consistently, and we've been monitoring this over the last 20 to 30 years, is that only 20% of people who are at risk of Huntington's disease actually decide to go on and have a predictive test. And that's where genetic counselling comes in, and it's really important to help people to make the decision that is the right decision for them at the time that they come and see somebody. And so predictive testing is only possible through clinical genetics services with the appropriate genetic counselling. There are 23 genetic centres within the UK. But each of those has lots of satellite clinics, and so there will be clinics near to wherever you live that you would be able to go to. And it's also really important to say that being referred to clinical genetics doesn't mean that you have to have a genetic test. It is really just an opportunity to sort of think about weighing up the pros and cons and thinking about when might be the right time for somebody to have testing. And we follow an international recommendations for how predictive genetic testing for Huntington's disease should be carried out, and that means that there are a number of appointments that lead up to the actual blood test, and a number of appointments that follow on from having the blood test with the results. And the aim of genetic counselling predictive testing is to support somebody to make the best decision for themselves at the time and to ensure that they have the appropriate support afterwards. So what do we need to think about when we're thinking about predictive testing? So there are lots of things to think about, but particularly we know that people from HD families might have complex family situations. And there are some times where, we might offer predictor testing to people who are not at 50% risk, so they're not the person with a parent who is affected. There might be times when we would be asked by somebody in the next generation, when their parent has decided not to have testing, to think about predictive testing for them. And so we are able to see people in clinic at lots of different risk times, but obviously it would be much more complicated for us to do a test and to give somebody a result if they had a parent who hadn't been tested, because we have to think about the implications to that parent of finding out that they may be at risk of Huntington's disease themselves. So all of these things are open for discussion, but sometimes we have to be quite careful about how we explain things to people and how we manage families. So it's important for everybody to think about complex family situations and how to manage them. It's important to think about the psychological impact of that a result might have, whether or not it shows that someone has inherited Huntington's disease or not, and the sources of support that might be available to them. It's important to think about, actually, lots of people come to see us because, they just want to know, they want to have some certainty about whether or not they are going to develop Huntington's disease. And sometimes we are just replacing one type of uncertainty with another. So if we get a result and it shows that someone has inherited the HD gene, then we're thinking about, well, actually, what we can't answer is, when symptoms are going to start, what they're going to look like, how they're going to progress over time. And so sometimes that causes more sort of worries that we're not able to manage so well. And so it's important to think about that in advance and what somebody might want to do. Obviously, people often come to talk to us about family planning and also research participation. And it's important to say for both of those things that actually you can participate in research, or you can also have family planning options, whether or not you choose to have genetic testing for Huntington's disease. And so all of those things are things that we would be able to talk to you about before you come to have your predictive test, if that's what you decide to do. Lots of people are anxious about certain things, so employment, driving, and insurance, and again, sort of thinking about those more practical things rather than the emotional side. So there is an Equality Act that means that people should have access to support at work if they have a particular genetic condition or any other condition. But in particularly the Army and other armed forces may discriminate against people who have a positive gene test or a family history of Huntington's disease. So this is the answer to a parliamentary question from a couple of years ago, that candidates who are known to be carriers of the gene associated with Huntington's disease are not normally graded medically unfit for service. That is even if they don't have symptoms, but they've just had a predictive test or a family history. And so it's important to sort of think about those sorts of things when you're thinking about whether or not to have a test, and if that's going to be important to you. There are some sort of police services that also would take a similar approach. If it is right or if it is wrong is a completely different matter, and we can definitely, you could ask for my opinion on that on another day. Some people worry about driving and the DVLA. So if you have a predictive test for Huntington's disease and you are not symptomatic, you do not have to tell the DVLA about your predictive test. It is only if you become symptomatic from Huntington's disease that you have to let the DVLA know. And even if you are symptomatic, lots of people are able to continue driving for a number of years, but have to have a yearly review with somebody to ensure that there aren't any symptoms that would make driving dangerous. And the other thing that people worry about is insurance. So this really is only applicable to life insurance. So there is a code on genetic testing insurance, which means that people who have had a predictive test for Huntington's disease do need to declare the results of that predictive test if they are taking out life insurance of more than for a value of more than £500,000. And so that affects a small number of people, but it is something to think about when you are thinking about predictive testing. And we would often advise people to sort of think about taking out their insurance before they had their predictive test result, or at least to sort of seek advice on that before they came to testing. So to talk a little bit about family planning options, lots of people choose not to let HD influence their reproductive decisions, and that is completely valid, and it's always been completely valid to do that, and particularly when we're thinking about clinical trials happening now, and that that children of people who are born now will have another 30 years until they might be at risk of developing symptoms themselves, and sort of putting your faith in science is not an unreasonable thing to do, and everyone has to make the decisions that are right for them. But we are going to talk about today is pre-implantation genetic testing and having a test in a pregnancy. There are also other options like adoption, fostering, sperm and egg donation, surrogacy, that would also be available to lots of other couples who are having difficulties with family planning. There's a question mark by adoption because there are some agencies that are quite stringent in terms of checks for prospective adopters, in terms of knowing whether or not somebody is at risk of developing a particular sort of illness, or if there's cancer in the family, or other sort of health difficulties, then couples might be restricted in being able to adopt, and so it's important to sort of seek advice locally. And we will always say that the best time to talk about family planning is well in advance of wanting to start a family, particularly if you want to think about things like pre-implantation genetic testing or testing in a pregnancy, because there are often a number of steps along the way that are important, and it always adds time onto onto people's decisions. So what is pre-implantation genetic testing? It's a bit like IVF. So couples who have fertility difficulties might have IVF, which means that for the female partner, that they will have some fertility drugs which stimulate the ovaries to make lots of eggs. Those eggs will be extracted and then put together with the sperm to make the very first cells of the developing fetus. At a very early stage, some of the cells will be removed in order to do the genetic test, and then only embryos that don't carry the genetic expansion for HD would be available for reimplanting. And so that sounds brilliant, and it is for lots of couples, but it's not without its own difficulties. The largest at the moment is that there are long waits for pre-implantation genetic testing within the NHS at the moment. So it's usually around a year from the time of referral until somebody is able to have an embryo transfer. It isn't always successful like IVF. The success rates for pre-implantation genetic testing are around 40% for each cycle of PGT. So there are some couples that I've worked with who have had 3 cycles of PGT and still haven't had a successful pregnancy, whereas I also know some couples who have gone through the whole process once and then they have had a healthy baby, which is fabulous. So it's important to go into it with your eyes open. There are some NHS funding criteria that people need to be eligible for pre-implantation genetic testing, and so it's important to think about those things in advance. And also it's really important to know that currently NHS funding is only available for the first successful pregnancy. So whilst up to 3 cycles will be funded, only the first up to the first successful pregnancy will be funded. And so if a couple did want to have more than 1 child, then they would have to think about, what their options might be for a second pregnancy, whether that is a private PGT cycle or what a number or what they might want to do. And so it's important to think about that in advance too. And so PGT isn't possible for all couples, and there might be some occasions when somebody is referred to us when they are already pregnant. So it is really still important for us to be able to offer a test to people in a pregnancy if they would want that, and if they would be would consider ending that pregnancy if it were affected with a risk of Huntington's disease. And so we do that via 2 different types of tests. So ideally, we would do that at a very early stage of pregnancy via a CVS or a chorionic villus sample, which is an invasive test that takes a little sample from the placenta, which has the same genetic material as the developing fetus. We do that early on. It does come with a small risk of miscarriage. And we usually get a result within a week to 10 days. So that would allow a couple to make a decision about whether or not they wanted to end the pregnancy if it were at high risk of Huntington's disease. And we would be able to do that at a very early stage. If a CVS doesn't work for any reason, or if a couple presents to us much later in a pregnancy, then we would arrange an amniocentesis, and we can do that from 16 weeks of pregnancy. It has a slightly lower risk of miscarriage, but only very slightly lower. But obviously, the longer a pregnancy continues, the more challenging it is for a number of different reasons to end that pregnancy. And so it's important to sort of think about that as soon as, in advance as well. There are two different ways of us being able to do testing. And so, as I said before, having a prenatal test or having PGT and thinking about family planning options isn't only available for people who have had a genetic test themselves and know that they are at high risk. It is also possible to have some of these options in some circumstances if you've chosen not to have a genetic test yourself, but have a family history of Huntington's disease. And so there are two different types of testing that we would offer. So direct mutation testing for a HD gene expansion is carried out where we know that a parent is affected or has had a positive presymptomatic test for HD, and that will tell us what the gene status of the fetus or the embryo is. But it is possible for us to offer exclusion testing where the person at risk hasn't had a predictive test, and exclusion testing doesn't give away the status of that parent. And so just to try and explain that, and it is complicated, but just to try and explain that very quickly, is that if we have enough DNA samples from people in the family, then we can look at markers along the chromosome rather than the HD gene itself. And so in this case, this granddad here has Huntington's disease, and when we look at his chromosomes, we can label them A and A. And this grandma here, she is unaffected with Huntington's disease, and when we look at her chromosomes, we can label them B. And so we know that this person who is at risk of Huntington's disease, he has a 50-50 chance of inheriting Huntington's disease from his father. He must have one A chromosome and one B chromosome. And so either in pre-implantation genetic diagnosis or in a prenatal test, we can look at the chromosomes of the developing fetus, and we can see if they have inherited an A or inherited a B. And if they have inherited an A, then that would be a high-risk pregnancy. And if they've inherited a B, we can be absolutely certain that they haven't inherited Huntington's disease. And so this can be really helpful for some couples who absolutely don't want to have a predictive test. And there are lots of reasons why somebody might not want to have a predictive test. But obviously it comes with its own set of challenges. It takes time to set it up. It requires samples from multiple family members, which means good communication between the family and between the genetics centres. And it also means that if the person at risk eventually doesn't carry Huntington's disease, then you would have gone through all of this testing unnecessarily, and you may even have ended a pregnancy that actually eventually would have ended up not being at high risk of Huntington's disease. And if you were doing it through pre-implantation genetic diagnosis, then you will have fewer embryos available for reimplantation, because you will be not implanting any that actually carried the normal copy of this granddad's chromosome 4. So why is genetics important for developing treatments? So we know that people carry the HD expanded gene from the time of conception, from the time of birth, and they don't develop any symptoms until some point during their lifetime. So they're completely unaffected with Huntington's disease until such time that they might develop symptoms and then be diagnosed with Huntington's disease. And we also know that brain cells are completely normally functioning until some time before symptoms start to develop, where they might start struggling, and then there is neurodegeneration later on. So it should be possible if we were able to support these struggling neurons and to sort of repair them in some way, that we at some time that somebody didn't have any symptoms of Huntington's disease, that we could prevent or delay the age of onset of HD. And because we understand what the gene does, and we also understand how genes are replicated and how the gene is translated and expressed, it should be possible if we had the right science to prevent the HD gene from being translated or transcribed and prevent it from making the faulty copy of the gene that causes problems within the cell. And so that technology hasn't been around for very long, but it is emerging, and it's just about trying to find the right technique for doing that, and the right way of getting it into people's bodies or brains in order to do that. And as you know, there's lots of clinical trials going on at the moment, and other drug companies that are trying to develop those exact molecules. And so it's really only in the last 5 years or so that we have seen clinical trials that are sort of gene-based. And so you will have heard about the Roche Generation HD2 trial. So that's an antisense oligonucleotide that binds to the RNA and prevents the mRNA and then the protein from developing. There are other types of ways that we might be able to do that. So something called CRISPR-Cas9 might change the actual structure of the DNA. There are PTC therapeutics have a drug trial at the moment of a tablet that should alter RNA splicing and prevent the faulty copy of the protein from developing. So lots of different approaches, and by understanding the genetics, we should be able to develop more approaches to that. You might have heard about Unicure. I think they announced yesterday that they've had some good results from their single gene therapy, which interferes with this here, so it stops the translation of the mRNA. And so, there are lots of different approaches, and it's about finding the right one. I said that I would talk about this, and I'm going to do it very briefly. So we know that the symptoms of Huntington's disease can be really variable between different people, even within the same family. We also know that if you have a certain CAG repeat size, that has a huge variability in when someone might develop signs and symptoms of Huntington's disease. And part of that might be due to environmental factors, but part of it might also be due to other genetic factors. And so this graph here shows the number of CAG repeats and then the variability of the age of onset. So you can see if you have 40 CAG repeats, you might develop symptoms in your mid-30s, or you might develop symptoms in your 90s. And so there is whilst the average age is around 60, there is a huge variability in when somebody might actually develop symptoms. And so the CAG repeat size itself only explains about 60% of the age of onset. And so what explains the rest? So I think there will be definitely some environmental factors, but through genome-wide association studies of thousands of people, 9,000 people with Huntington's disease, and knowing their age of onset, scientists have found that there are a number of different genes that might influence somebody having earlier or later onset of symptoms. And whilst individually these genes might not help with predicting age of onset for an individual, they might help us to understand certain targets for developing treatments or therapies that might delay age of onset. Lots of these genes are involved in DNA repair mechanisms. And as I said earlier, DNA repair might influence how many of those CAG repeat sizes can get packaged up and effectively. And so they're really important to understand. And you can also see that one of the genes that they looked at was actually Huntington's itself came up as potentially being a modifier. And so here is your CAG repeat size here. You can see at the end of this gene, there is a CAA and then another CAG and then a CCG. So the structure of the actual HD gene itself might give us some clues as to why the gene might be unstable. So most people have these repeat interruptions at the end of their HD gene, but there are different structures that exist in different people. So the commonest one, just like this, has a CAG repeat, then a CAA, then another CAG, and then the CCG and the CCA. But there are some people who don't have the CAA, and there are some people who don't have this CCA. So all of this is very complex, and I'm not going to go into it very much, but it could be that rare structural changes in the gene could affect the age of onset, or how likely the gene is to expand in future generations. And that's something that scientists are working on at the moment, but again, isn't there for individualized risk at the moment. And it might also tell us something about somatic instability, which you might have read about. So that is where actually everybody with, if you have a blood test for Huntington's disease, it will give you two numbers of what your CAG repeat size is. And that doesn't change over your lifetime. So there is no variability really in the blood. But actually, from looking at brains, post-mortem brains of people with Huntington's disease, we can see that CAG repeat length is very different throughout different tissues of the body. And so it will be very much higher numbers than your blood at a later stage of disease. And so again, being able to look at the DNA repair genes might be really important for helping us to target that somatic instability and develop therapeutics. So I think I've got to the really complicated bit just at the end, and so I'm going to stop there. But really just to say that even though HD is caused by a single change in a single gene, the genetics is really complicated. It should be that anyone who is at risk of Huntington's disease has choices and should have access to skilled professionals to help them to understand what those choices might be. There are 23 UK genetic centres available to help. And the understanding of the genetics is leading to development of potential disease-modifying therapies. A really good place to go to if you want to learn more is the HD Buzz website, and lots of the therapeutics stuff that I was talking about. You can go to the CHDI Foundation, where they had a conference in February this year, and all of the talks from all the scientists are available there. Thanks very much to everyone for listening.